Hi, today I'm going to continue the next session in systems biology, and this is from one of the book that is called Basic Concepts in Medical Systems Biology. Here I'm presenting section 1, where it is basically covering the pathways biology approach to medicine, or in another word, you can understand the role of signaling pathways for therapeutic purposes in broader sense. That means to understand the role as well as how to and where to apply the drugs for targeting in order to diagnose the specific disease. So systems biology provides a new approach to studying, analyzing, and ultimately controlling biological processes. Biological pathways represent a key subsystem level of organization that seamlessly perform complex information processing and control tasks. The aim of pathway biology is to map and understand the causal effect relationship and dependencies associated with the complex interactions of biological networks and the systems. Drugs that therapeutically modulate the biological processes of disease are often developed with limited knowledge of underlying complexity of their specific targets. Considering the combinatorial complexity from the outset might help in identifying potential causal relationship that could lead to a better understanding of the drug target biology as well as provide a new biomarkers for modeling diagnosis and treatment response in patients. So this is one of the representation of post-genomic medicine loop where you need to understand literature, data mining, clinical data, modeling, experimentations. Before or after modeling, you can perform the experimentations. Now, this is one of the simplest example of signaling pathways, how a signaling pathway is triggered. And this is called TCR mediated MAP kinase signaling. So this is the T cell receptor location, then intracellular signaling molecules, for example, the RAS, RAF, and MET, and finally the target or downstream signaling molecule which conveys the information or transmits the information, signaling information to the nucleus. Then afterward, the cells undergoes differentiation or apoptosis or proliferation or growth. Now, the same thing has been simplified in another way, which you need to understand is the signal level, then TCR or receptor level, then intracellular signaling molecules, and then target proteins. And finally, you could expect the response depending on the cellular response of ERK. Now, modeling or and formalizing the dependencies, the main cellular processes, conceptual representation of the main cellular processes that occur inside the cell. Signaling cascades receive external signals from the environment, either by binding to extracellular receptor or something in another way. The signal is then propagated through a signaling cascade that involves the sequential phosphorylation of several proteins, leading to the gene activations. Gene regulatory networks control the transcription level of genes, and genes are then transcribed into RNA molecules, which are subsequently translated into proteins. And these proteins are involved in cellular functions. Some proteins are enzymes that are involved in the catalysis of metabolic reactions, and metabolic networks obtain energy and carbon from external sources using internal conversion steps. The internal metabolites can be used for cellular growth or converted into byproducts that are excreted by the cells. Their concentration level can also influence the gene regulation. And this is the combined network representation for signaling network, gene regulatory network, and metabolic network. Here you can easily see how all these three networks are connected with each other and or dependent on each other. The different symbols or shapes are representing. For example, this one is signal, then channel, receptor, protein, gene, RNA, and metabolites. Now it comes to signaling network. Signal transduction is the process for cellular communication where the cell receives external stimuli from other cells and from the environment. It affects most of the basic cell control mechanisms such as differentiation, apoptosis. The transduction process begins with the binding of an extracellular signaling molecule to a cell surface receptor. The signal is then propagated and amplified inside the cell through signaling cascades that involve a series of trigger reactions such as protein phosphorylation. The output of these cascades is connected to gene regulation in order to control cell function. Signal transduction pathways are able to crosstalk, forming complex signaling networks. So here, this is FGFR4 mediated signaling, which you can see the pathways affected or activated by the activation of FGFR4 or FGFR1, 2, 3, or and 4 are in more than one. And this receptor has also been used for drug targeting purpose for different human diseases. And the location of the receptors as well as signaling molecules you can see. This is extracellular and then transmembrane and then cytoplasmic and nuclear regions. Now it comes to gene regulatory networks. Gene regulation controls the expression of the genes and consequently all the cellular functions. Although all of the cell functionality is encoded in the genome through thousands of genes, it is essential for the survival of the cell that only selected functions are active at a given time point. Gene expression is the process that involves transcription of the gene into mRNA, followed by the translation of it to protein, which may be subject to the post-translational modification. The transcription process is controlled by the transcription factors or TFs that can work as activators or inhibitors. The transcription factors are themselves encoded by the genes and subject to the regulation, which altogether forms the complex regulatory networks. And this is the simplest form of for the representation of gene regulatory network, where you can see the chromatin accessibility, transcription factors, and their targets, microRNAs, long-range chromatin interactions, and then protein level, post-transcriptional regulations, and finally, it goes to different biological processes. Now it comes to metabolic network. So metabolism is a mechanism composed of a set of biochemical reactions by which the cell sustains its growth and energy requirements. It includes several catabolic and anabolic pathways of enzyme catalyzed reactions that import substrates from the environment and transforms them into energy and building blocks required to build the cellular components. Metabolic pathways are interconnected through intermediate metabolites forming complex networks. Gene regulation controls the production of enzymes and consequently directs to the metabolic flux through the appropriate pathways in function of the substrate availability and nutritional requirements. So here, this is the example to show the metabolic regulatory network. This is the metabolic regulatory networks, and the other things are connected with metabolic networks. So you should not consider that metabolic network is not connected or affected with the other signaling networks or gene regulatory networks or some other external factors, but it is connected with many things in addition to the metabolic components. Now it comes to the requirements for modeling, and that is computational modeling in system biology. So due to different properties and behavior of the biological networks, they usually require different modeling features. And for instance, features such as stochasticity, multi-state components may be important for signaling, but not for metabolic networks. A summary of the major modeling features required by these networks could be seen easily here In this network, computational modeling in system omics data, biochemistry, cell biology, and physiology data sets, large scale data, then quantitative and qualitative data, then genomics analysis, and dose response curves analysis. Here you can take the help of networks, microarrays, NGS, and in this case, you can take the help of time courses, action potentials, patch lamp clamp, and then statistical analysis and dynamical modelings. And finally, this is the complete map or view for system biology. Now it comes to the network visualization. So once you process all these preparation for the mathematical modeling, you also need to analyze before and after simulation the network properties, and presentation is the most important part in case of system biology. These are the different ways to represent the views of the biological networks, and you can also represent in this way or this way, depending on which gives you better results or conclusive results and easy to grab. Now it comes the Cytoscape, which is one of the most common software which is used for network building and visualization. So these are the large number of way of presentations for the network. So this is one way of presentation, another way of presentation, and then another, and this is another way, here another way. So a number of, this is not the limited number, but there are more than 50, 100 way of visualizing the network by using this single Cytoscape software. And Cytoscape also needs to have some apps which are easily installed in Cytoscape. Now it comes to view the miRNA's networks. So you can also view or represent the most complex network or biological network, that is miRNA network, in this way. And go next, it comes to the analysis, topological analysis. When you have complex networks which you cannot easily analyze or visualize with your open eyes, then in that case, you need to analyze the network properties or topological properties. For example, degree, connectivities, and betweenness. All these things you need to analyze for the complex networks in order to make some conclusive remarks. Now, this is another way of presenting the most complex network. And then you can also present in this way the master regulatory genes or proteins. For example, these circles are representing one central protein here, and these things could be considered as the master regulator in the network. For example, this represents the YAP1 and WWTR1 TAS stimulated gene expression. Then it is nuclear transcription pathways. Then it comes to the transcriptional activity of SMAT2, 3, and 4. So you can think in multiple ways. Then it comes to modularity and hierarchies. So despite its great complexity, the cell is organized as a set of connected modules with specific functions. Taking advantage of this modularity can help you to alleviate the complex burden and facilitating the model analysis. Compositionality is a related concept, meaning that the two modeling blocks can be aggregated together into one model without changes to any of these submodels. This property can be of a special interest for applications in synthetic biology. While modularity represents the horizontal organization of the cell, living systems also represent vertical organization. Molecules, cells, tissues, organs, and organism populations and ecosystems reflect the hierarchical organization of life. A modeling system that supports hierarchical models and different levels of abstractions will cope with the models that connect vertical organization layers using top-down, bottom-up, or middle-up approaches. And these are the concept for modularity. This is what you see in the form of modularity, and this is hierarchical way of representation. Now it comes to the multi-state component. There are different way or different perspective in which you can talk about multi-state, and mostly in signaling you need to understand the multi-state. But I'm saying I'm presenting few views of multi-state components. Some compounds may have multiple states. For example, the protein may be modified by phosphorylation. This is a very common case in signaling process. The state of a protein can affect its functionality and consequently the reactions in which it participates. Therefore, different states are represented by different entities. However, a protein with n binding sites will have 2 to the power raised by n possible states, which results in a combinatorial explosion of entities and reactions. To avoid this problem, a suitable modeling formalism should consider entities with internal states and state-dependent reactions. And these are one way of the representation. Now it comes to a spatial structure and compartmentalizations. So on its lowest level, the cell can be seen as a bag of mixed molecules. However, this bag is compartmentalized and requires transport processes for some species to travel between the compartments. Furthermore, in some compartments, including the cytosol, the high viscosity, slow diffusion, and amount of molecules may not be sufficient to guarantee a spatial homogeneity. A spatial localization and concentration gradients are actually important mechanisms in biological processes. Now, it comes to the next part where you can see, for example, this is one way where you are easily seeing the things in different compartments, escape through membranes, flux imbalances, and you can go to the cell level, you can go to the cell organ levels, you can go at multiple levels. Now, this is the grand view or presentation for the compartmentalization. So the how the complex systems or any living organism systems could be represented at different compartments or layers over the layers, that is multi-layered representation. And this is one of the finest figure to represent the entire organisms at different level or compartment and different complexities, classification, many more things you can do it. So now we move towards the qualitative and quantitative analysis. This is very important. Once you move to perform the modeling and simulation and analysis of the network, you also need to analyze during simulation process how the temporal dynamics of the proteins or genes are changing, and that is like the concentration or expression level of the genes or proteins with respect to time. That is called temporal dynamics, and this plays crucial role. The temporal dynamics plays crucial role in controlling the cell fate decision, that is apoptosis, proliferation, cellular growth, dif differentiation, and many more things. So what is dynamic simulation? Dynamic simulation allows the prediction of transient behavior or sustained behavior of a system under different conditions. For each model, the particular simulation approach depends on the type of component included, which depend on the nature of the involved interactions and also on the available information for the characterization. In regulatory networks, genes are activated and deactivated through the transcription machinery. Due to their complexity and the lack of kinetic information, the transcriptional details are usually not considered. Instead, genes are modeled by using discrete variables and that change through the discrete time points. This is the simplest simulation method and requires models with a little detail. A spatial signaling cascades, especially signaling cascades, are triggered by a low number of signaling molecules. Therefore, it is important to take into consideration the inherent stochasticity in the diffusion of these molecules. A stochastic simulation is a common approach for simulation of signaling networks. This approach requires the attribution of probability functions for each reaction in the model. Metabolic reactions, on the other hand, comprise large quantities of metabolites. Therefore, their behavior can be averaged and modeled by continuous variables governed by deterministic rate laws. This requires significant amount of experimental data for the estimation of kinetic parameters. Now, the same thing, you can see how these things, dynamic simulation, could be represented as a graphical or in the pictorial way. So this is the one of the finest way to represent the entire dynamic system which you are going to perform for or include for the simulation purpose. Now, this is the final workflow for the representation of your modeling or system biology approach for the dynamic system. Now, standardization comes. There has been a lot of approaches or trial from different researchers and research organizations to develop some kind of a standard way for developing more or in a more standard way the systems biology field. But so far, everyone has failed. And system biology markup language, that is SPML, is one of the oldest approach or trial which badly fell. Then it comes to SBML and then SBGN. There are then another one is like called cell ML. So you do not need to bother much, but you can also prepare yourself all these things. And these are some of the examples for the uh, a trial for standardization of system biology approach, but they are not successful. So now it comes modeling formalism. So formalism, you can understand from the term itself, have been used to model biological systems in part due to the diversity of phenomena that occur in living systems and also due to the multidisciplinary needy of the research teams. Biologists may be more familiar with mathematical modeling, and computer scientists may be religious to their computational formalism of choice. The dichotomy between the mathematical and computational models has been discussed a lot in multiple ways. But what you need to understand, you need to have the background of basic mathematics and background of really good coding or computational programming, and then you also need to have in-depth knowledge of biological sciences. Now it comes to different types of networks or modeling approaches. One is called Boolean system, where you use 0, 1, 0, or -1 also sometimes. So I have also discussed the Boolean system where in the last previous lectures in computational biology and bioinformatics. But here I'm just summarizing. The Boolean system saves your time. You can perform the mathematical modeling and simulation of much bigger network with respect to the others. And it is quite easy. But in terms of experimental correlation, it, it does not fit that much with respect to other approach. So now it comes to Bayesian network, which were introduced by Pearl 1988. They are a special type of probabilistic graphs. So instead of going so much details, because I have discussed the Bayesian things, so you need to understand there is some probabilistic approach for simulation or modeling and simulation of biological network. And here you can see one way of representation of Bayesian network. Now it comes to the Petri nets. This has been quite common in use for a few years, 3, 4 years ago, or let's say 7 years ago, that some people were using Petri nets, where it is quite easy that it is graphical user interface, which can easily give you the mathematical equation. You just need to click and add protein or add some metabolites or add some genes in your way of understanding, for example, as you can see here. Then in the end, you can develop or generate the mathematical equation for your mathematical modeling and simulation purpose. So that could save your time, but it badly failed. Now it comes to the process algebra or basic mathematical science, which you need to know. These are some of the common things which are used or which are required for mathematical modeling and simulation. Now, constraint-based models for the cellular metabolism is assuming that the cells rapidly reach a steady state. These models overcome the limitations in lack of experimental data for parameter estimation inherent in fully detailed dynamic models. They are based on stoichiometric and thermodynamic and design capacity constants. Instead of a single solution, they define a space of possible solutions representing different phenotypes that comply with the constraints. The simplicity in this formulation allows its application to genome scale metabolic models comprising thousands of reactions. So this is much easy, but you need to understand the entire system, and this is one way of representation of constraint-based modeling, and this is mostly used for metabolic network modeling and simulation. Now, the same thing that could be seen in the complete details of pictures. Here, this is annotated genome, then reaction data, then you need to make some matrix, and then flux, and then you go for some phenotype prediction by using some different kind of steady state analysis, thermodynamic constants, capacity constants, and optimization process, and finally you make the conclusion on phenotype predictions. Now, differential equations, this is one of the most reliable on prediction, which gives one of the most reliable prediction, and that is differential equation, where you use ordinary differential equations and for mathematical modeling and simulation of the biological networks, especially for the signal transduction pathways and gene regulatory networks. Now, there is another thing called rule-based modeling. So this comprises a recent approach for the problem of multi-state components in biological models. In rule-based formalism, the species are defined in a structured manner and support multiple states. The reaction rules are defined as transformations of classes of species, avoiding the need of specifying the one reaction for each possible state of a species. This high-level specification is then automatically transformed into a biochemical network with the set of species and reactions generated by the specification. This kind of formalism is implemented in BioNetGen, which generates the ordinary differential equation, and also you can try it in PetriNet. How rule-based modeling works, you can see here. Rule-based systems are deterministic, rule-based systems are modular, and then rule-based systems can be adaptive. Rule-based systems can be used to simulate the complex behaviors. Rule-based systems have limitations also. Now, interacting machines, this is quite simple. Model of biological systems as state machines. Now, it allows multiple levels of hierarchy to capture different levels of detail in biological systems. Then model concurrency through the definition of parallel communicating state machines. So, for today,